My name is Harold Ott. I'm founder and CEO of IVIVA Medical, a startup company in Boston that tries to develop new solutions for patients with end-stage renal disease, patients with kidney failure. I'm excited to be here today to share progress on our program, share our vision, and share our enthusiasm to participate in the KidneyX community, where our team is one of the lucky winners of the KidneyX prize. And this will certainly catalyze a lot of synergies between the researchers, the research teams, and hopefully help us bring our technology to patients. At IVIVA, we're trying to develop living therapeutics to cure kidney failure. Kidney failure is a silent epidemic. It affects many patients, about 1 million patients in the US alone. And once you reach a stage where your kidneys are not able to remove toxins and remove water from your body, your quality of life suffers and you do require replacement therapy. What that means is that 25,000 patients of this 1 million receive a life-saving transplant after waiting for 5 years on average, while 100,000 of these patients die each year. That means 275 patients die per day waiting for a cure. So this is certainly a need. As a physician and surgeon, I'm very familiar with this, seeing patients with renal failure and seeing how they suffer from the treatment as well as from the disease and from its consequences. My journey in the field of kidney engineering started several years ago when I had done basic science research on trying to build functional tissue from cells. And after one of these publications went out, a third grader sent me this email, saying, "Dear Dr. Ott, I have kidney problems. I take a lot of medicine to help this, but since you figured out how to make a kidney, I can be able to take less medicine. Have a good night." As a physician, I was aware that of the impact of the disease in general, but this was a very palpable message telling me we really need to figure something out here. The person that sent me this email is doing very well. She received a kidney transplant at age 12, and she just finished her first year of college. But if you look at the end-stage renal disease, at the kidney failure field as a whole, you realize that once you develop renal failure, your options are quite limited. Life evolves around treatment of the disease. You spend 3 hours a day, 3 to 4 hours a day, 3 times a week attached to a hemodialysis machine. You get placed on a wait list, and if you're lucky, you receive a donor kidney, but only 1 in 5 patients on that wait list receives a donor kidney. So roughly 500,000 patients depend on hemodialysis, and 25,000 patients receive a transplant. And once you do receive a transplant, your life doesn't quite return to normal. You require lifelong immunosuppression that can lead to many complications. Your organ will last for about 10 years, and eventually you'll need repeat transplants. And currently in the US, there's only roughly 1,500 repeat transplants done. So while I'm excited for this patient that emailed me, I also recognize that she is doing well for now, but she does need a solution and does need us to work on this. When you start to work with stem cells and understand biology and physiology, our body is quite amazing, and that's something that to this day fascinates me when I do surgery and when I stand in the lab and work with cells. Cells are so much better than machines in performing high-level functions. And a good example to highlight this is that in your kidneys, about 1 ounce of cells does the same job as a 200-pound piece of equipment, what you see here on the right. That's roughly the size of a ping pong ball of cells being able to perform the same as a dialysis machine that's the size of a standard household refrigerator. So the question, though, is how can we leverage these cells and how can we use these cells as biologic building blocks to build functional therapies, build advanced therapies, and build organ replacements? I had done a lot of basic research since starting my surgical training in trying to deliver cells to improve patients' organ function. But at the time, we did not have technology to derive these patients from cells. And that world changed dramatically when Professor Yamanaka developed the technology together with others that allows us to reprogram cells from a patient's blood sample. What that means is that today we can actually create biologic building blocks from a simple blood draw and drive them to become the necessary cells to rebuild functional tissues such as heart muscle, kidney, or lung tissue. While this progress made this field a whole lot more relevant and allowed us to think about how to transform these cells into therapies, I recognized, and so did many others, that cells really need three-dimensional structure to function at an organ level. What that means is that the cells in your body are the smallest living building blocks, but they form structure that is organized by what's called an extracellular matrix. What you see in this picture is the smallest functional unit in your kidneys. You have about 1 million of those, the size of a grain of sand, and they're called glomeruli. So these are the little balls where blood flows through and the first urine in your kidney is created. And the colored cells that you see on there are called podocytes. These are cells that sit on this three-dimensional structure and are able to perform their function because they're very, very well organized. So our body has figured out ways to not only provide the cells, the building blocks, but also put them in a three-dimensional context that allows them to function at this higher level. It's a little bit like an office building with people living and functioning in it and being able to perform work. So for us, the engineering hurdle was not necessarily only to derive the cells, but also develop a scaffolding system that allows us to bring these cells into the correct three-dimensional context. What you're looking at here looks quite simple, but what it is a layered structure that takes this ball that you saw in the picture before, flattens it out onto a flat surface, and allows us to seed cells on a very large surface that can easily be blood perfused to replicate some of these biologic functions that provide you with water removal and toxin removal every day of your life. You see that this person is holding this structure because it is roughly the size of a donor organ, roughly the size of a human kidney, and the pipes that you see attached to it are vascular conduits, basically biologic types of conduits, of pipes that can be connected to the recipient's vasculature, very similar to a donor organ, and function in the background after implantation. The long-term vision here is to really build these devices from patient-derived cells, meaning that patients will not require immunosuppression, not require any supportive, continuous monitoring and supportive care, and can undergo repeat procedures once these devices stop functioning over time. Our team is very excited to join the KidneyX community. This is an innovation accelerator, as it says on the logo, but that is absolutely true. The KidneyX community brings together scientists that have been in this field for decades, have developed some of the foundational technologies that we're building on here, and together work towards a future where hemodialysis is a short-term bridge and organ wait lists are a distant memory. So with that, I want to thank you and KidneyX for listening, for your support, and look forward to the years to come for this technology to advance. Thank you.